Welcome to the next episode of IBD Essentials. It's a great pleasure to introduce my co-host, Florian Rieder today, but before he is going to introduce himself, I would just like to thank you for watching so much of our previous videos. If you like us, you can like us. If you have interesting topics that we should address, if you would like to have some different topics, please just get in touch with us. You can write everything here on our page and we will change it. Florian, we have been long-term friends and colleagues. We have met, I'm not sure, but you will know probably much better, more than a decade ago. We have been working on several programs together. We had very, very nice projects. Now you are head of IBD in the Cleveland Clinic, and it's very impressive work you're doing there. I have been your guest last year, and I was really, really impressed with the breadth of the work you're doing there. But today, you will introduce yourself first, but today I think our main topic will be fibrosis and everything around fibrosis, because all those who haven't met Flo so far, Florian Rieder is the Mr. Fibrosis. I think he devoted almost all his career for the research on fibrosis, and he moved it really from the bench or early bench to the bedside, and is now getting a lot of attention in the recent months. Florian, perhaps you introduce yourself. Great pleasure that you are here with us.
Yeah, thank you, Axel, and thank you for having me as co-host of your series, which has been very successful. And thank you to everyone for dialing in and listening to us. So I'm looking forward to the next 20 minutes. I'm Florian. I'm a gastroenterologist from the Cleveland Clinic. I'm leading the research enterprise at the Cleveland Clinic in gastroenterology, hepatology, nutrition, and global inflammatory bowel diseases. I have, where Axel and I met, a bit of an unusual path. I trained in Germany before I came to the United States. I met my wife in the United States, stayed there, and then retrained from the beginning and graduated not even 10 years ago.
Fast career. But for me, because we started a long time ago, also when you were already in the US at ECHO, we made projects together, and it was always a great pleasure to work with you, because you are a great mentor, you're a great speaker, you have great ideas, and what I really like a lot, you are really able to connect people, bringing people to work together and to identify the best of the individuals and bring this to a huge mass. And this was very impressive when I was visiting your unit, and the breadth of everything. So it's not only IBD, it's nutrition, it's basic science, it's clinical science, and they all work nicely together. And before we really go into fibrosis, one thing I also liked a lot, you were really, as you mentioned, global IBD. So it's not the Cleveland Clinic in Cleveland, but you have the Cleveland Clinic in London and Abu Dhabi and Florida. And you have with this also a huge network of different settings and different patients. So is this really helpful? Or is it more like a burden because you have to oversee so many different places and different people?
Yeah, thank you. And thanks, Axel, for your kind words. I like a lot working with people, as you point out, and I get very excited about it. So at the Cleveland Clinic, it's a very unique enterprise where we have multiple hospitals in Cleveland, multiple hospitals in Florida, and then we have one in London and one in Abu Dhabi. We are currently creating one global IBD unit. So we discuss the patients together virtually four times a month. We have research projects together. We have quality projects together, clinical standards, and this truly one global unit. And I think there's huge potential. Sarah El-Wali, for instance, who you will also interview shortly, is in Abu Dhabi, Charlie Murray, and then Asad Ur-Rahman. We work very closely together to improve patient care.
Yeah, no, this is really exciting. But let us move to fibrosis. And how did it come that you were choosing fibrosis? Because this is exciting, but it's also connected to one of our, let's say, unsolved clinical problems. If we just consider Crohn's disease, strictures are still something we struggle. We end it with surgery or with dilatation. drugs are in the pipeline, but for a long, long time, and when you started, I think there was no drug even considerable in the pipeline, at least for me. Why did you start to work on fibrosis? What was exciting you?
Yeah, so it's a good question. So I'm generally, with my personality, drawn to problems. I would like to solve problems. I want to do this all the time. And when I started seeing IBD patients, it doesn't take long for Crohn's disease patients to present with obstruction and strictures. And it bothered me that we have no way to treat it and we have no way to prevent it. So when I started, regardless of which anti-inflammatory therapy you use, the patient eventually, close to all of them, had obstruction in a tertiary center. And so I dedicated a large part of my career to solve that problem. And at that time, when I started, I was laughed at several times because it was felt that it's not current and we have to treat inflammation and everything else will go away. But it turned out it's not the case. And we are now on the brink of having the first anti-fibrotic therapy.
Long way to this. So you really started basic science in the lab, but also initially, long time ago, you were working on endoscopic dilatation, and this also brings up one of the major problems. We have no real classification of stricturing disease, and when we will end up discussing clinical trials, we also need tools to measure and to evaluate drug treatment. So you led several consortia, and this is also, again, probably due to your talent. You brought people from all over the world together to think and to end up with classifications and methods to measure this. So probably you would like to start a little bit and tell us the background, how you developed this, how you brought the people together, and what type of classification we can ultimately use.
Mm-hmm. So we started, as you mentioned, very basic in the laboratory. So we first developed tools to measure success of therapies and predict success of therapies. Cellular systems, primary human cells, animal models, tissue models, and so forth. This was one big important pillar because I think we need to have a pipeline of potential mechanisms we can target. A second pillar was the clinical development path, and you referred to the clinical development path. So it bothered me a lot. We found all these targets in the lab, we published them, and it creates positive attention. But at the end of the day, nothing ended up with patients because there were no regulatory pathways. If there's no regulatory pathway, industry does not engage and invest in a clinical trial. And when I started thinking about that, it was striking to me that there were about 20 different definitions of what a stricture is on imaging alone. There were about a dozen different definitions for what strictures on histopathology and several different on endoscopy. And the first thing, which sounds simple, when I graduated from fellowship, I formed a group to just define what is a stricture. And on imaging, we defined that it is a combination of wall thickness, luminal narrowing, and then pre-stricture dilation. So when the patients have this luminal narrowing and the feces keeps on coming, there's a pressure zone upstream and then the bowel balloons up, which is probably an indirect marker of this stenosis phenomenon. It's likely, we think, linked to patient symptoms. And we harmonized also the definitions for endoscopy. And then we defined what constitutes improvement, and you referred to clinical trials. And the regulators have now clearly mandated you have to have an improvement in symptoms and an objective marker, not unlike for luminal disease. And the objective marker is either endoscopy or imaging. And to measure stenosis on endoscopy is very difficult, and the reliability has been shown not to be high. And so at the moment, the regulator opinion is to use imaging as the second piece of it. And then to walk the path towards registration, we then properly develop these tools, and this is not particularly sexy or flashy work. This is grinding through regulator-mandated steps. And we developed a patient-reported outcome tool. So how does the patient feel? How does this manifestation impact the patients? And there are three phases. So you do a concept elicitation. You ask the patients, how do you feel when you have stricturing disease? You collect 20 or 30 of them, and then you have certain themes. Then you generate items, so you create a draft instrument, and then you ask the patients, got cognitive interviews. If they understand the questions correctly, then you have a draft instrument. We have this draft instrument now, and we have a prospective validation for it that we're going to report this year. And the instrument performs, in fact, quite well. The second pillar is imaging. And just like the definitions that were not existent before, to measure something on imaging sounds very simple, but really is not. So you need to develop training programs, you need to develop reliability exercises. We identified items that you can reliably measure, so these are fit for an endpoint, and we understand what is called the minimal detectable change. If a patient gets better and they go a stricture that is 10 centimeters long, goes to 9.9 centimeter, you can argue, can you actually measure that? Yeah. But we understand now which improvement you can actually accurately measure. And we did that for MRI, and we did that for CT, and we are doing it for ultrasound. So we are now in a very good spot, in fact, to test novel anti-fibrotic therapies in stricturing disease.
So you already mentioned the first two pillars. Before we go to the third pillar, I would like to go back to the first two, because you really brought some important points. It's again MDT approach, because the GI cannot do anything alone. We need surgeons, we need the radiologists or the ultrasonography people, and you brought in one or two important players. I think our top player are always the patients, so we need to work with the patients. But you also brought in the regulators, the FDA, EMA, because nothing will help us if the regulators will not approve certain new treatments and new drugs. And for this, we need these tools. And this was really groundbreaking work. You mentioned it may not sometimes disheartening, but the patient point of view was the first pillar. I think this is extremely important. The second pillar is something which I feel is very complicated for certain people in certain areas, because in some countries you have good MRI, in some you have CT, in some countries CT is not really doable because of radiation, in some countries ultrasonography, you even have the possibility to see and talk to the patient, and you see the functional effects. So how do you get this sorted out? Because you are having a global program. You need to look at different countries, and in certain countries, one option will probably not be available. So how did you get this sorted out? How did you...
So this is an excellent point. So we started with MR, and when the first trials, and we can talk about the trials in a minute, when the first trials started testing anti-stricture therapies, it turned out exactly what you were saying. So if you're bound to MR, this is somewhat a restriction, particularly in less developed countries, where this imaging technique is not available, or the standard of the scanners is not high enough for a clinical trial. For that reason, we developed the CT tool, and actually both are interchangeable. So the measurement accuracy is close to identical between the two. So even within clinical trial, you could, in theory, use these both techniques interchangeably, and you have the same accuracy. Ultrasound needs more work, so we have shown that you can reliably measure strictures if you have one operator and 4 central readers. But in clinical trials, you will not have one operator, but you have one operator per center and central readers. And so we are currently starting a study to test multiple readers in the same patient and using central reading to get it fit for clinical trials. But I think at the moment, ultrasound is emerging and is very promising, but I think a lot more work needs to be done. But the trials that are ongoing are restricted largely to larger centers that have these MRI capabilities.
Just on a side, not our major focus today, but the whole world is talking about AI, and these, especially MRI and CT, seems to me prone to use AI also to make it easier with central reading and probably to help the investigators. Any thoughts on this?
Yes, so we have within our consortium several programs about artificial intelligence reads. One is to detect stricture volume in collaboration with a partner called Multiland. They have IT tools to not only measure stricture length, pre-stricture dilation, but the actual volume of the tissue. We have a virtual biopsy using radiomics, which is an AI imaging technique, and multiple other programs to do exactly that. I would also consider it in the infancy. It is experimentally interesting, but until a regulator accepts an endpoint, takes a long time, and when you put artificial intelligence on top of it, it takes even longer. So from a regulator perspective, we stick at the moment to very simple measurements like stricture length, ball thickness, pre-stricture dilation, and do AI experimentally for early development.
Yeah, this is very important, and I think this is true for all the other aspects. The regulators are really tough, and they want simple endpoints, reliable endpoints, and all the other points will be added later on.
But I think one thing that you mentioned, which I think is critical to highlight, in this consortium building these endpoints, we call it Stenosis Therapy and Anti-Fibrotic Research Consortium or STAR Consortium, truly interdisciplinary, truly MDT, with surgeons, radiologists, clinical trial endpoint developers, gastroenterologists, in collaboration with regulators, patients, and then the big third pillar is industry. So we would not be able to do this without industry. We have about 20 industry partners that then invest in the clinical trials, invest in the tools. It can only work together, which I think is a highly enjoyable process. In the beginning, everyone needs to understand each other first, and then it needs to speak their language. But it's a highly enjoyable process.
But you need probably a person like you to bring all these people together. I think the STAR Consortium is something which is really having a lot of impact all over the world, and you brought the important players together, and now you are not really close, you're in the midst of it. You started the first clinical trials, you presented some of the data. Perhaps you will just summarize where we are standing now and give us a glimpse what will be coming probably in the very near future.
Yeah, so we're very excited because we now have the first trial completed with an anti-fibrotic therapy, but maybe I go 2 steps back. The first attempt was with a drug called spesolimab, which was an anti-IL-36 receptor antibody. There was is a fibrotic pathway that was designed for stricture and Crohn's disease patients and gave us the first idea about the regulatory landscape in that space. Boehringer Ingelheim was running it at that time. Unfortunately, it did not come to fruition for business reasons. That was a critical step. And then the second step, which is being presented at ECCO in 2026, is the STENOVA study. So this is an ALK5 inhibitor. The chief profibrotic cytokine across organs is transforming growth factor beta. And there's one critical kinase for signaling at the receptor called ALK5. The challenge with inhibiting it in cancer, for instance, and other fibrotic diseases, are systemic side effects, cardiac valves, systemic autoimmunity in cancer. And so the drug called ontunizertib is a locally delivered drug with first-pass metabolism to the liver with low to undetectable systemic levels, but high mucosal levels. The population are patients with terminal stricture, non-critical, meaning they are not expected to have to undergo surgery or balloon dilation over the next 12 to 24 weeks. And the symptom burden was manageable. And then they were randomized to 2 doses of ontunizertib, high dose, low dose, or placebo, on top of standard of care. So these patients came in already on an anti-TNF or an anti-IL-23 or anti-1223, and then had the drug added on for a duration of 12 weeks. At inclusion, they had MRI, then the symptom assessment and endoscopy, and at 12 weeks, at MRI, symptom assessment and endoscopy. And to be honest, when starting this trial, very advanced patients, very severe strictures, 17 years disease duration, 85% have been exposed to biologics. All patients had small bowel disease location. All patients had strictures. And if you run a trial in this population with a biologic, you can essentially say, already this is likely going to fail. So my expectations for this to be successful were quite modest. But in fact, we saw very nice signals with the high dose of ontunizertib in an early development phase 2A study.
Wonderful. So there is probably a very promising data and studies ahead. just probably before we stop, we also heard a lot of noise about the TLA1 inhibitors because they seem to have dual activities, so anti-fibrotic but also anti-inflammatory activity. And you mentioned earlier this was your starting point, inflammation and fibrosis may work together. So can you just share your expectations on the use of TLA1 inhibitors, because there are several around there, so it seems to be exciting.
Yes, so this is a big new kid on the block, so anti-TL1As. We have 3 phase 3 development programs for UC and 3 phase 3 development programs for Crohn's disease. And at last count, multiple more companies that have clinical developments either ongoing or starting in earlier stages. The promise of anti-TL1A is that in phase 2 studies, very strong signal, in fact comparable to upadacitinib, comparable to anti-IL-23s. So far in the patients that have been tested, it's early days, but no significant safety signals either. And the big promise of this drug is a potential anti-fibrotic effect. And what became clear over the years is that treating inflammation alone does not prevent or reverse stricturing disease. And in fact, over the last decades, we saw an increase in the need for surgery for patients with stricturing disease, not a decrease, which you would expect, with more and more potent anti-inflammatory medications coming. And TL1A is an amplifier, so it has multiple sources and it has multiple cells that receive a signal. Clearly a lot of inflammatory cells receive the TL1A signal, but also fibroblasts directly receive it through a receptor called death receptor 3 or DR3. And in animal models, inhibiting TL1A inhibits fibrosis, and inhibiting TL1A only on fibroblasts, TL1A signaling, also inhibits fibrosis. So I think the promise here is not only treating inflammation, but also treating inflammation-independent mechanisms of fibrosis.
Florian, this was really great, and this was probably also a great stop, but you already started several new podcasts, because I think this is something where we could just have a whole series. And I also liked that you mentioned trials in ulcerative colitis. A lot of us always think that fibrosis will only occur in CD, but we know that in ulcerative colitis it also has a very important role. So it was really, really wonderful. I hope that you will have also exciting guests in the future, but I would also like to have several podcasts with you together, because this was really amazing. Thank you so much.
Yeah, thank you for having me, and thank you everyone for dialing in.
And to our listeners, once again, if you like us, please like us. If you have ideas for new topics, just send the topics, make notes below, and we will try to address whatever seems to be appropriate. Thank you, and hoping to have you back for further episodes of IBD Essentials very soon.